Armando Hasurigan, Biology and Medicine videos. Please make sure to subscribe, join the forum and group for the latest videos. Please visit Facebook, Armando Hasurigan. Please like, and here you can also ask questions, answer questions, and post some interesting things, including your artworks. You can also change the quality settings to the highest one for better graphics. Now, in this video, we're going to talk about oxidative phosphorylation, also known as the electron transport chain. Before I begin, please note that the actual oxidative phosphorylation process, the electron transport chain, is not fully understood, and new stuff keeps coming up in the science world. So yeah, just remember that. So some things might be left blank, but know that this is the overall sort of picture in a way. So we begin here inside the cell. Here are the two mitochondrial membranes. Here's the cytoplasm, the intermembrane space, and the matrix inside the mitochondria. And so if this were the outer mitochondrial membrane, the other will be the inner mitochondrial membrane. Now, the electron transport chain consists of four important structural proteins known as the four complexes: complex 1, complex 2, complex 3, and complex 4. Also in the oxidative phosphorylation process, we cannot miss out the one important enzyme which creates the ATP, known as ATP synthase over here. ATP synthase is sometimes known as the complex 5, but for now we'll just call it ATP synthase. Now, the electron transporters, the four complexes, each have names. Complex 1 is known as NADH dehydrogenase. Complex 2, succinate dehydrogenase. Complex 3, cytochrome C oxyreductase. And complex 4 is known as cytochrome oxidase. And lastly, complex 5 is known mainly as ATP synthase. Okay, now the two important substances to begin the oxidative phosphorylation process are the electron carriers from glycolysis, from the preparatory step, and the Krebs cycle. And the total amount of electron carriers from each of these processes we have are 10 NADHs and 2 FADH2s. So 10 NADHs and 2 FADH2s are the total amount of electron carriers we obtain from glycolysis, the preparatory step, and Krebs cycle. Okay, knowing this piece of information, we have to understand what electron transport chain is and what oxidative phosphorylation is. Essentially, what electron transport chain is essentially where an electron is being transported through this chain. This chain is complex 1, 2, 3, and 4. And so, these electrons, where do they come from? They come from the electron carriers, the 10 NADHs and the 2 FADH2s, which were obtained from glycolysis, preparatory step, and Krebs cycle. So the electrons are obtained from these electron carriers, and then they travel through the electron transport chain, where the electrons will then, their final destination will be oxygen, which will help reduce oxygen to form H2O. So oxygen is known as the final electron acceptor. And then during this process, as well as when the electrons are moving through this electron transport chain, hydrogen ions are being pumped out. And with these hydrogen ions, ATP is made. So that was just a brief overview. Now let's begin with NADH, the electron carrier NADH. And let's look at only one NADH and its influence and what happens to it and its electrons through oxidative phosphorylation. And we begin with NADH through complex 1. Some important structures within complex 1 is a flavin mononucleotide, FMN, and also a series of iron centers, as well as an iron sulfur center, N2 over here. Before we continue with complex 1, another important protein structure which floats in the inner mitochondrial membrane is ubiquinin, also known as coenzyme Q10. And its purpose is to carry electrons as well through these different complexes, because it is a mobile protein, whereas the complexes are stationary. So let's go back to complex 1, NADH dehydrogenase. Let us follow the electrons and the fate of one NADH plus H molecule, which is an electron carrier. So NADH dehydrogenase will oxidize NADH plus H to form NAD. And through this process, it will obtain two electrons. The two electrons will be firstly given to flavin mononucleotide, FMN here, and then the two electrons will pass through a series of iron centers like so, where it will stop at the iron sulfur center. And next what happens is that the two electrons will create a proton gradient. It will bring in two hydrogen ions from the matrix, and then will bind to ubiquinin. And as it binds to ubiquinin, it will reduce ubiquinin to make ubiquinol. And so ubiquinol has two hydrogens. So we write it up as QH2. And also during this process, the electrons, as it travels through, will pump 4 hydrogen ions from the matrix into the intermembrane space, like so. Now, ubiquinol is essentially carrying the two electrons, but it's not as electrons, it's just as hydrogen. So ubiquinol will travel through the inner mitochondrial membrane with the two electrons, right? And it will travel through, it will not associate with complex 2, but it will associate with complex 3. Now, let's look at complex 3. Complex 3 has a few important subunits, 3 important structures within it. One is called cytochrome B, the other is called Reiki iron sulfur proteins, and the other one is called cytochrome C1. The most important structure, however, is cytochrome C, which is a mobile protein in the intermembrane space and is attached to the complex 3. Complex 3, cytochrome C oxyreductase, will essentially oxidize ubiquinol to ubiquinin, and it will essentially steal the 2 electrons from it. And it will pass the 2 electrons to cytochrome C. So now this cytochrome C has 2 electrons, let's just say. And also during this process, as the electrons are passing through, complex 3 will also pump 4 hydrogen ions from the matrix into the intermembrane space. Next, cytochrome C will travel through the intermembrane space and attach to and bind to essentially complex 4, the cytochrome oxidase. And it will attach to one of the subunits of complex 4. Complex 4 consists of 3 main subunits, 1, 2, and 3. Simple enough. And as it gives the 2 electrons to complex 4, cytochrome oxidase, will then, with the 2 electrons, reduce oxygen, half an oxygen, which is essentially 1 oxygen molecule, with 2 hydrogen ions to form 1 water molecule. And also during this process, cytochrome oxidase, complex 4, will pump 2 hydrogen ions from the matrix into the intermembrane space. Now I hope you understand why this is called an electron transport chain, because the electron is transported between all these complexes, where it will arrive at oxygen finally. And so we say oxygen is the final electron acceptor. And remember, these electrons only came from one NADH. And so now, if we calculate all the protons pumped from one NADH, we have got 4 from complex 1, another 4 hydrogen ions pumped up from complex 3, and another 2 from complex 4, giving us a total of 10 hydrogen ions, 10 protons being pumped from the matrix into the intermembrane space through one NADH molecule. And also this creates 1 water from complex 4. And so what do we do with these 10 hydrogen ions which are in the intermembrane space? Well, it will get fed, we can say, it will go through the ATP synthase, complex 5, to produce ATP. So let's have a look at ATP synthase. ATP synthase consists of many subunits. It consists mainly of 2 complexes. It has a F0 complex and an F1. Let's look at the F1. F1 consists of alpha and beta subunits, a gamma subunit, and also a epsilon subunit here. And the alpha and beta subunits is what actually produces the ATP. That's what ADP and organic phosphate binds to. F0 consists of 10 C subunits, an A subunit, 2 B subunits, and a delta subunit down here. Now, all these different subunits we don't actually have to memorize, we just have to know their functions. So ATP synthase will make ATP when hydrogen ions pass through this complex. And it's actually 4 hydrogen ions that have to pass through to make 1 ATP. Now, to make ATP, there are actually many ADPs, adenosine diphosphate, and inorganic phosphates inside the matrix, right? And so when 4 hydrogen ions pass through the A subunit of F0, this will cause the C subunits of the F0 to rotate. When it rotates, it will essentially phosphorylate ADP to form ATP. So 4 hydrogen ions passing through will create 1 ATP here. Therefore, because we have 10 hydrogen ions in the intermembrane space from 1 NADH, this means when 10 hydrogen ions pass through the ATP synthase, it will produce a total of 2.5 ATP. Just think about it. So 1 NADH will finally make 2.5 ATP from 10 hydrogen ions.